For the main presentation, I'll hand over to Heather Stark, Global Segment Marketing Manager for Biopharma at Oxford Nanopore, for an introduction on how Nanopore sequencing works.
Thank you, Diana. I'll briefly explain Nanopore technology and discuss how Nanopore sequencing solutions can be pivotal for biopharma, because what you're missing matters. So the cost of drug discovery and development is risky and exorbitantly expensive, with only about 10% of therapies ever receiving approval. Advances in genetic analysis have made an essential tool, and having the most comprehensive view of your genome, transcriptome, and epigenome can enhance the probability of success. Nanopore sequencing can expedite the discovery process by accurately uncovering previously undetected genetic information, leading to the identification of novel mechanisms of action, new biomarkers, and reducing risks in therapeutic development. So how does Nanopore sequencing work? The animation on the left demonstrates the fundamental technology behind Nanopore sequencing. Our sequencing devices feature an array of nanopores set within a membrane that facilitates the concurrent sequencing of multiple DNA or RNA strands. During the library preparation, an adapter with a motor enzyme is attached to the template. This motor enzyme propels the reaction when a current is applied, guiding a single nucleic acid strand through the nanopore. As the strand moves through the nanopore at a velocity of 400 bases per second, it disrupts the ionic current, generating robust, reproducible signals that we translate into base calls. This technique is not constrained by the limitations of synthesis-based sequencing technologies. It can directly sequence native DNA or RNA, enabling it to avoid PCR bias while maintaining natural modifications such as methylation, without the need for additional treatments such as bisulfite. Nanopore sequencing is adaptable in terms of read length. Read length ranges from 20 bases all the way up to megabases, sometimes in a contiguous read. It's also remarkably fast, delivering real-time sequencing data. This rapidly enables unique nanopore-specific techniques like adaptive sampling. Additionally, we offer compact, efficient devices and flow cell options to suit any laboratory need, all without substantial investment in equipment. I mentioned we're developing applications to support genetic analysis across the gene therapy value chain. Rapid whole plasmid validation using nanopore sequencing allows for accurate detection of mutations, not only in the transgene, but also in the vector backbone, whilst revealing contaminants that could negatively impact production. AAV vector quality control with Nanopore assesses integrity of viral mutations from ITR to ITR, identifying aberrant mutations, potential truncations, chimeras, and other contaminants. Accuracy in these critical quality attributes is essential for ensuring efficacy of viral vectors in gene therapy applications. And lastly, due to our long native reads that span insertion sites, Nanopore sequencing provides deep characterization of transgenic CHO cell lines, for example, or your animal models, to verify gene edits, correct delivery orientation, and even to resolve unwanted edits such as incorrect duplications or inversions. So today we concentrate on AAV sequencing. I'd like to give you a streamlined overview of our protocol. Our AAV protocol is a comprehensive one-day process that takes you from sample to report, utilizing our epitome analysis workflow, AAV-QC. Maximize the recovery of high-quality, full-length AAV genomes, we suggest beginning with the PureLink extraction kit, followed by a DNase treatment. This approach does not require a self-annealing step so you can transition immediately into library prep. The library prep process involves NPrep, NIC repair, DA tailing, and concludes with the direct ligation of sequencing adapters, either ligation sequencing kit for individual samples or the native barcoding kit for multiplexing. After loading the samples onto the flow cell, sequencing results can be obtained in as little as 1 hour. In summary, Nanopore sequencing offers a powerful solution for research or QC labs studying recombinant AAV, providing accurate, efficient, and comprehensive sequencing results. By leveraging the power of Nanopore technology, QC labs can enhance their quality control process to ensure safety and efficacy of AAV-based therapeutics. Thank you for your attention. I'll now hand it over to Daniel and Bastian for their presentation.
Thank you, Heather, for the introductions, and thank you, ONT, for inviting us. So in our talk, me and Bastian will present how we evaluated adeno-associated virus genome sequencing on Nanopore platform and what kind of new insights we can learn with this new way of QCing AAVs. So I will start with the short introduction to AAVs. I'm not a virologist, so this is very simple, basic information. So AAVs are naturally occurring viruses infecting human and some other primates. In fact, 30 to 60% of human population has already been exposed to wild-type AAVs, and then infection is usually asymptomatic. They have a single-stranded DNA genome of approximately 4.7 kilobases, and the characteristic feature of AAV genome is the presence of inverted terminal repeats flanking the open reading frame of wild-type AAV, and these are really like a hairpin secondary structure made of palindromic sequences that can be sometimes tricky to work with. So AAVs appear in several serotypes that have different tissue tropism. They can infect dividing and non-dividing cells. And then once in the cell, AAV genome has the capacity to integrate into the host genome in the human's specific location on chromosome 19, and then be sort of in the latent state in the presence of helper virus or co-infection. This AAV genome can be rescued and then undergo the replication cycle and then be packaged into the capsids and released to the environment. So in recent years, AAVs have gained a lot of attention, specifically in the gene therapy field, for a number of features listed here that make them a really promising platform for gene delivery in the gene therapy. But to be used in gene therapy, they have to be modified in a way that wild-type AAV genome is replaced with a therapeutic cassette. And these recombinant AAVs, then they don't have the ability to integrate into the host genome, but rather they exist as the episomal DNA or extra chromosomal DNA. Then these genomes can perform their therapeutic function, such as they can replace or silence mutated gene, overexpress therapeutic protein, and generally they have a long-term gene expression profile. What's a little bit challenging about recombinant AAVs is the coding capacity, which is 4.7 kilobases. So it's important to optimize the therapeutic sequence so it's efficient, and in some cases it might not be simply possible to for very large genes to put them inside the AAV capsids. But how are AAVs, or recombinant AAVs, actually produced? So standard AAV expression system comprises of 3 plasmids and the packaging cell line. So the plasmids are transgene plasmid carrying the therapeutic sequence cloned in between the ITR sequences, and we have a helper plasmid and replica plasmid, and they really help with the packaging, and they supply for the replication and capsid proteins. These 3 plasmids are then co-transfected into the packaging cell line, usually hex cell line, and are expressed, and then the transgene can be packaged inside the capsids, and then the vectors or AAVs can be released to the medium where they are harvested and purified, and then later used for the phenotypic rescue studies. So it looks fairly simple, and technically we should get AAV capsids only containing the transgene sequence, full-length transgene sequence flanked by ITRs, but we don't actually know it for sure until we put it to the test or employ some QC methods that can actually assess the AAV cargo. And it's important for the safety and development of recombinant AAVs. So recombinant AAV should contain only the transgene sequence and be free from contaminants, such as the fragments of the host genome or packaging cell line genome, or fragments of helper plasmid, replica plasmids, or the parts of the backbone. And there are some QC methods, conventional QC methods like gel electrophoresis, qPCR, that are a great starting point when the new designs or iterations of AAV are produced, but they offer really limited insight. They don't give us a full picture. On the other hand, DNA sequencing or genome sequencing is a much more comprehensive method, and it can inform us about the specific sequences of the fragments within the capsid and inform us about the abundances, and when paired with the long read sequencing, also give us information about the integrity of a transgene, and we can also detect chimeric genomes, so fusions between the transgene and the replica or helper plasmid, and so on. So it happened that we had the Nanopore sequencing on site at AstraZeneca, so we decided to give it a go and basically evaluate this workflow. So in the next slides, we will present a pilot experiment that could be an inspiration for other labs. And it was really to see what kind of data we can get with this workflow, how can we analyze it, and what sort of insights we can then get from it. So I will start with the wet lab part of the evaluation. So in our pilot experiment, we used in-house produced AAVs with relatively short transgene, 2.6 kilobases long, coding for EGFP, and this was really to clearly distinguish the transgene sequence from the human background or human genome. Additionally, we used lambda DNA spiking to test for the DNA's efficiency. And DNA's treatment is to digest or remove any DNA that could be present outside the capsid or could be carried over. And we do this treatment just to make sure we don't have any background. And we only sequence what's inside the capsids. Then these samples were lysed with the proteinase K treatment and heat. For DNA extraction, we opted to use Zymo DNA clean and concentrator columns. We have a good experience with these columns, and they also have a little bit lower size exclusion point, so they can retain fragments longer than 50 base pairs. And then we did a QC. We always do the QC before library prep and sequencing. This is a really important step for any NGS work. For AAV samples, we used Fragment Analyzer. This is a capillary electrophoresis instrument that can pretty accurately size the DNA fragments. We used NanoDrop to read the absorbances ratios and learn about the possible contaminants like protein or ethanol contaminants. And then we used Qubit, also very standard for Nanopore library preparation. Qubit just gives more accurate quantification of the DNA. So this slide shows the QC results for our DNA. We had five different samples with iterations of sort of lambda DNA spiking or digestion. We were happy with the purity of the DNA. We actually got quite a lot of DNA. So potentially, yeah, we probably used too much AAV capsids or genomic copies. for this experiment. And in the bottom part of the slide, you can see the fragment analyzer results with a very sharp peak around 2.44 kb, which is for the full-length transgene sequence, and then two smaller peaks that technically shouldn't be there, 900 bases long peak and around 5 kb long peak. By the way, alkaline agarose gel is also a good way to QC AAVs, but in this case we couldn't see this shorter peak, 900 bases peak on the standard agarose gel. And what we really kind of want to QC is the smearing of the peak. If the material is intact, it should give or if the genomes are intact, they should give sharp peaks. If we see a smearing or some additional peaks, this is usually a good indication that the DNA is already fragmented before the library prep and sequencing. For the library preparation and sequencing, we used Nanopore protocol for AAV sequencing, which is based on native barcoding kit, chemistry 14. And it's pretty simple with entry per step, barcode ligation, pooling, adaptive ligation, and so on. There are some third-party reagents needed to perform this library preparation. For our, we used 70 nanograms of AAV sample, AAV DNA, genomic DNA per sample, and additionally we added extra control, which was linearized plasmid, a transgene plasmid. So in this way, we can at the same time sequence AAV and QCR plasmid, do the whole plasmid sequencing. We loaded entire library. We got really good pore occupancy plot, and actually we sequenced overnight. After 17 hours, we got 3 million reads, which was enough to do the analysis, more than enough. And actually, we stopped the sequencing after 17 hours. So this also can be easily rewashed and used for next sequencing for other AAV samples with different barcodes. So it really helps to get the best value for the flow cell or the sequencing. And now I will hand over to Bastian, who will talk about dry lab results.
Thanks, Daniel. From the wet lab into the dry lab. So now we'll talk a little bit on how we assess the rAAV sequencing in silico. Once we get the data off the sequencer, typically we are interested in checking for two specific categories of issues that can arise. First of all, we want to check whether or not we're packaging any undesired material. Ideally, our capsids contain only the ITR to ITR sequence. What we want to avoid is packaging any helper or repcap plasmids, any plasmid backbone from the AAV plasmid, or any host genome from the packaging cell line. As Daniel mentioned previously, typically we use HEC for this. The other category of errors that can arise are not from packaging itself, but rather from the content of the sequence. And that would be things like truncations or other sequence deviations. Think about SNPs or other variants, short indels, and something that some, particularly due to the nature of the ITR to ITR region, something that can arise is chimeric concatenars, which is also an undesired outcome in many cases. Our workflow combines both the epitomy, public epitomy workflow from Oxford Nanopore. On top of this, we have created some of our own custom tooling, some of our own custom scripts that work either with the output or in concert. I should note that we use the standalone version of the workflow. You can use this through Minno. But since we run most of our analysis on high-performance compute clusters, we typically just get the workflow itself and run it on the cluster. The way that the workflow generally works in a simplified manner is we take the sequence material that comes off the Nanopore machine, and that contains, at this point, unknown sequence to us. It could be anything from helper plasmid, host genome, AAV plasmid, ITR to ITR or backbone, or from the RAAV plasmid. The workflow then aligns this sequence data to a combined reference. The combined reference contains the plasmids, so the AAV helper and RAAV plasmids, the 4 versions of the ITR to ITR region. Why are there 4 versions? Due to the nature of the ITR being a terminal repeat, it can assemble itself in various configurations, so-called flip, flip, flip-flop, etc. And finally, the host genome. The aligner minimap will place each read on any or none of these particular references, and then we will create the report that comes from the Nanopore pipeline, and we use the output to do some custom analysis, typically using some implementation of R or Python scripting. The way that we handle this is more or less on a per experiment basis. If the experiment is interested in a particular aspect of the QC, we have the flexibility to just take the output from the pipeline and modify our analysis to their needs. And then we assess our data in 3 broad categories: contamination, completeness, and orientation. Here is some sample output. This comes from the ONT pipeline. Let's start on the left-hand side. We plot the number of reads against the quality score. So this is a Phred-encoded quality score. If you're not familiar with the system, on the x-axis, a value of 10 is roughly 90% accuracy, a value of 20 is roughly 99% accuracy, and a value of 30 is 99.9% accuracy. Our median here lies at around 15. This is slightly below what was expected of the flow cell in this case, since this is a R14 chemistry, so supposedly around Q20. However, for our workflow here, the coverage of the region of the reads themselves more than makes up for the slight drop in quality. So due to the sheer number of reads that we get off the platform, we can easily correct for spurious errors. On the middle plot, we see the number of reads plotted against the read length in kilobases. 2 peaks stand out. The 2.6 kilobase peak is our expected ITR to ITR region. The 5 kilobase peak is a control that we had in this experiment, and in this particular experiment had a linearized plasmid control, which is 5 kilobases long. What we are paying a lot of attention here to particularly is that, first of all, we do see peaks that we expect, usually the ITR to ITR region. Secondly, we pay attention to the level of noise, i.e. shorter reads, what is the proportion of shorter reads, and are there any other peaks that stand out? On the right-hand side, similar to the middle plot, this tells a story of how much yield we get in terms of bases as a function of the read length. What you want to see here is sharp drops around your expected read length. So in this case, we have our first sharp drop at 2.6 kilobases, another sharp drop at 5 kilobases. Now, when we assess packaging contamination, we want to make sure that the vast majority of our ITR, of our sequence data is originating from the ITR to ITR. Here in this plot, we are looking specifically at only the pAAV plasmid. So at this stage, we are not yet sure that the reads are specifically in the transgene ITR to ITR region. They could be still from the backbone at this stage. However, we will later look specifically how much of the data is within the ITR to ITR region and within the backbone specifically. We see some other peaks here. We have some helper and some unmapped, and a larger chunk of the reads get assigned to either the host HEC packaging cell line or RepCap. A question that often comes up at this stage is how much is acceptable? I personally don't have a great answer for this. There is no one true measure of how much packaging contamination is acceptable. It really depends on the experiment, on the target, therapy, etc. So for us, we typically hand off the data to the biologics team, and they make decisions based on other variables, whether or not this is an acceptable level or not. The other interesting part on this data is we can assess the completeness. In an ideal scenario, all our reads would be perfect ITR to ITR reads, essentially sequencing the entire ITR to ITR region from start to finish. However, in real life, we can see things like shortening of the ITR regions. We can see truncation hotspots. In some cases, the transgene is truncated at a specific hotspot, and this is an obviously very detrimental QC issue and the problem for the therapy vector. So what we do is we look at the start and end positions of the alignment. So each individual read, we collect start and end positions, and we assess, does it start within the ITR? Does it end within the ITR? Or is there a specific part within the transgene where it ends? This looks like this. After we're done assessing, we have on the x-axis here, the position of the ITR to ITR part of the transgene plasmid. This is in 50 bins. So this is not base pairs. This is just taking the 2.6 kilobases and dividing it up into 50 equally spaced bins. On the y-axis, we have the alignments. So a bar plot of the number of alignments that either start or end in this position. And the color represents the strand. Does it align on the plus or minus strand? Now, this is a good example. Most of our data either starts or finishes at the very ends of the region. This is exactly what we want to see. If you were to see a peak or a large number of starts or ends that are not within your expected ITR regions, then that could highlight an issue here. In addition to looking particularly at the completeness of the ITR region, we also pay attention to chimeric alignments. What we have here is a circular representation of, right now there are two plasmids visible. So we have in light blue the RepCap plasmid, and in all the other colors the AAV plasmid. The gray represents the backbone, the mulberry color represents the ITR regions themselves, and the lime green is the transgene. We can specifically look at two things in this. So this is a circus plot. In the middle ring of this, so the rings that are right beneath the actual schema of the plasmids, we see coverage plots. What immediately pops out here is that virtually all of the sequence coverage originates from within the ITR to ITR region. There is a mild but not a concerning drop as we leave the ITR specifically and enter the transgene. So there is slightly less coverage of the transgene region as compared to the ITR region. But in my opinion, that is not concerning. That is not a concerning level of coverage drop. virtually no other parts of either the backbone or the RAAV or helper plasmids are covered by a significant amount of reads. In the innermost circle, we now see ribbons that highlight chimeric alignments. So these are alignments that start either at one end of the plasmid and originate or end up on the other end, which is natural because this is a circular plasmid. If it's still circular when we're sequencing it, the aligner doesn't really know that it's a circle. Minimap doesn't deal with circular alignments. So what it will flag the alignment as a chimera. This is completely normal. So if you see chimeric alignments that are on the 5 or 3 prime end of one of the references and end up on the inverse, that is to be expected. We see some chimeric alignments here that are hopping in between, so alignments from either the rAAV or the helper plasmid or the AAV plasmid to any of the other plasmids. The level of this is very low, and for the most part, these are looking like false positives from the alignment. So this is something that is not concerning at all. I have a, let's say it more failed example. So what we see here is a similar plot. I have taken out the helper plasmid from this plot as it's not adding much information. This is a sample where there was not only significant, essentially there was a complete loss of the transgene. You see in the mulberry regions, again, our ITR regions, our lime green would be the transgene. However, there is virtually no coverage present. So essentially the transgene is lost. There is significant coverage of the backbone. And in addition, there are 3 hotspots or 3 somewhat covered regions in the rAAV plasmid, which also have chimeric alignments into the AAV plasmid. This could be, once again, this could be false positives from the aligner. We see that a lot of these alignments, these chimeric alignments, do go and end up in the ITR regions. Repetitive regions are notoriously difficult to align through. So if these are shorter reads, it could be an alignment artifact or it could be a plasmid fusion. It's good to keep that in mind. And still, we report these. Finally, we have the AAV structure output. I typically take this with a grain of salt. So as you can see by the color scheme, this comes once again from the ONT pipeline. Why do I say I take it with a grain of salt? The pipeline is configured on our end to be very strict with what it considers a full SSAAV and a partial SSAAV. As soon as a few base pairs of the ITR region are shaved off and not fully aligned through, the pipeline is configured to consider it as a partial SSAAV. However, in practice, it is not clear how much of a difference that would make. In practice, I would personally consider this to be somewhere around 90%, 80 to 90% full SSAAV. The pipeline further infers other types of AAV structures. We see backbone contamination, only ITR region unknown, etc. These can be very helpful when trying to deduce the efficacy of your pipeline, trying to understand what has worked well, what has not worked well. In summary, we have found that recombinant AAV sequencing on Nanopore is not only simple and inexpensive, it also delivers high quality and very useful data for us. We definitely use the AAV-QC pipeline from the Oxford Nanopore team. In my opinion, it provides a really good starting point for the AAV quality control. Sometimes we have to build on it when there are specific experimental questions, but often it answers the core concerns straight away. We could detect in some experiments levels of packaging impurities, specifically host and plasmid DNA, and we could often confirm that the transgene is still intact, and sometimes even detect chimeric transgene. Finally, we do expect some read truncation, and there are various steps in the protocol that may shorten your ITR ends, that may look as though there is a quality issue. However, this is, in my opinion, somewhat artifactual, and we think it is important to establish internal quality thresholds, what you and your team consider to be acceptable levels of shortening or acceptable levels of contamination. This is not only my and Daniel's work, so we would really like to acknowledge some other parts of the AstraZeneca team, members like Fiona and Julie, as well as Mariam, who have spent a lot of time, a lot of effort to develop our AAV QC platform in-house. Thank you for your time.